All right. Okay, so again, good morning. Thank you for joining us. My name is Gian Smith, and I am the StreamWatch Program Coordinator at the Watershed Institute. And today we are doing the Bacterial Action Team training for Session C, which is the last of our 3 5-week sampling sessions that happen throughout the summer. So let's dive in. A little bit about what we'll be going over today. We'll start off with a brief introduction of the Watershed Institute and StreamWatch as a whole, and then a little bit more specifically about the Bacterial Action Team, which is one of 3 teams within the StreamWatch program. Then we'll talk in a little bit more detail about harmful algal blooms, what they are, what causes them, what to look out for, and how to identify them. We'll look at our visual assessment with a tool called Survey 123, which is online. And that's the part where you might want to have your phones available so that you can practice going through the form on your phone, which is what you will use in the field. And then we'll go into detail about how to collect water samples, the transportation of those samples and delivery of them to the Watershed Center. And then I'll touch briefly on how we do the analysis in the lab and what happens with that data once we've done the analysis. All right. So the Watershed Institute works to protect and restore our water and natural environment in and around central New Jersey. And we do that through 4 main pillars, the first of which is conservation. So if you've ever been to the Watershed Reserve, and if you haven't, I certainly recommend coming to visit. We have just under 1,000 acres of preserved land at the Watershed Center. Lots of trails over 7 miles of trails that you can walk, including our newly finished boardwalk trail. So this area is used for a lot of research. We do deer population studies and impacts on the environment through deer grazing. We have a lot of university students that come and do their theses using our preserved land. Preserving the land itself also helps to regenerate groundwater. And so in that way, we're contributing to water quality and water quantity for people who live in this area. That's a lot of times where we get our drinking water. And we also use our reserve for lots of educational purposes. So if you happen to visit during the summer, you'll see that we have lots of summer camp participants around, lots of kids. And but that is not just over the summer. Over the course of the year, we have lots of schools that come and visit. And we also, our educators also go out to schools and other programs to give education about the environment, why it's important, and why and what we're doing to preserve it. And then we have kind of our education for adults, as I like to think of it, or our advocacy program. And our advocacy staff take the things that we learn on our reserve and the things that we teach to our kids, to our students, and bump it up to a level where we can speak with municipalities and small governments and even the state government to help to push along regulations and ordinances that will help to protect and preserve water, clean water. And finally, all of that is supported by our science team, of which I am a member, and now so are you. And our science team is highly involved with a lot of the research that I was mentioning on the reserve, but we also do StreamWatch, which is our longest running volunteer water quality, or longest running science program, and it's our volunteer water quality monitoring program. So within StreamWatch, where there are 3 separate teams. StreamWatch began in 1992 with the Chemical Action Team. So Chemical Action Teams, affectionately referred to as CATs, monitor water chemistry on a monthly basis. So they go out on the 3rd weekend of each month to measure these water parameters: temperature, nitrate, phosphate, chloride, pH, turbidity, which is the clarity of the water, dissolved oxygen, and conductivity. And so that is our longest running team, over 30 years of data collected by those volunteers. Then we have the Bacterial Action Team, which is what you're joining today. The Bacterial Action Team monitors bacteria populations weekly from mid-May to the end of August. And we're using, in this case, E. coli as an indicator species for fecal contamination. And we're also acting as watchdogs to keep an eye on harmful algal blooms, which we'll talk much more about in a little bit. And finally, we have our biological action team. And this team goes out once a year in the spring to monitor macroinvertebrate populations. Macroinvertebrates are insects and snails and mussels and worms and things like that that live on the stream bed. And we know from previous research through other organizations that they have different pollution tolerance levels. Each organism has different pollution tolerance levels. So looking at the combination of organisms that we find, we can get a longer term, we can get a snapshot of kind of a longer term snapshot of the water quality, because that water quality needs to be sufficient to sustain the life cycle of those organisms. So if we can find certain organisms that have low pollution tolerance values, we know that that water has been healthier for longer. So those are the 3 StreamWatch teams, and we'll talk a little bit more about the bacterial action team. So we, as the Watershed Institute, started off as the Stony Brook Millstone Watershed Association, covering, let me use my little pointer here. I don't know if you can see this. Yes, okay. This Millstone watershed in central New Jersey. In about 2017, 2018, we expanded to include this lower portion of the Delaware watershed. So now our coverage area is this whole area in central New Jersey, which you can see in this little inset over here. And then this map, I realize, is a little bit hard to see at this scale, but these are our actual monitoring locations for the bacterial action team. So what we do is we send volunteers out to 2 or 3 of these sites every week to collect data. Let me turn the pointer off and keep going here. All right, so the expectations, I just want to get those out of the way here. First and foremost, we want to make sure that you're keeping yourself safe. The easiest, well, one of the first ways to do this is to make sure that you are going out with a partner, or at least letting someone know that you're heading out into the field. So please make sure that you're doing that. If there conditions at your stream are seem unsafe in any ways, you do not need to continue monitoring. And things that might make a site unsafe include high rushing water, unsavory characters, potential for flash flooding, a harmful algal bloom if it in a really extreme case, especially if DEP has already been there and notified that there are high levels of toxins there. You do not need to continue doing that. None of the data that we're collecting here is worth risking your safety. So please, first and foremost, make sure that you're keeping yourself safe. Secondly, please take care of the equipment and return it at the end of the session, which in this case also happens to be the end of the season. We do use this equipment over and over again, and so it's important that it's being taken care of and we can get the full life of the equipment out of it. This one is important. We ask that you do monitor every Sunday for the next 5 weeks. It's unfortunate when we have gaps in data. So that's what you're committing to doing right now. If you know right now that you're not going to be able to monitor all 5 sessions, please shoot me an email and let me know, so that we can either make up a plan or have something, or reassign your sites. But yes, so you're right now committing to monitoring every Sunday for the next 5 weeks. You'll collect your samples after 8 a.m. and deliver them to the Watershed Center by 1 p.m. every Sunday. We'll follow the directions for the sterile sample collection, and we'll go through that a little bit later, but this is important so that we're measuring the bacteria that is in the water and not the bacteria that is in your hands or in the cooler, etc. And if you haven't done so already, please make sure that you complete your volunteer forms. If you have no idea what I'm talking about, please reach out to our volunteer coordinator, Eve Niedergang. Her email is here, and she can send you the digital copies of what she'll need to fill out. And I'll just put an asterisk here and say that if you are under the age of 16, we do require that you have an adult participating with you. So that means someone here today attending this training with you or watching it later if you're watching it later. And also going out into the field with you every Sunday. And that also means that we'll need their volunteer forms completed. So if you are under the age of 16, please make sure that you have an adult participating with you and that they have also completed their volunteer forms. Alrighty. So this is a 15-week sampling schedule for this year. And you'll sample every Sunday between 8 a.m. and 12 p.m. with a drop-off at the Watershed Center no later than 1 p.m. So in a nutshell, this is what the Bacterial Action Team does. The Bacterial Action Team is unique in StreamWatch because it is the most focused on protecting human and animal health and safety in the water. And that's why we do our sampling over the course of the summer from mid-May to the end of August, because that is when people and their pets are more likely to be interacting with the water in our area. So how do we protect human and animal health? We do that by measuring the extent of bacterial contamination in our waterways. The first step in doing this is to conduct a visual inspection of the water body for toxic cyanobacteria. And you do that, you as a volunteer will do that by doing your visual inspection and then reporting those conditions using the Survey 123 form that we'll go over in a little bit. We also collect water samples for E. coli and other, which we analyze for E. coli and other chemical parameters. And so your role in that is to collect the samples and then transport those samples, keeping them on ice to the Watershed Center for testing. And lastly, we measure water temperature, which you will do using the thermometers provided in the kits that hopefully you have collected. So here's what your weekly routine will look like. Because rainfall can have an impact on the amount of bacteria entering the waterways, we ask that you check local rainfall totals for the past 24 and 48 hours. If you happen to live close to your sites, which most of you should, you might know just offhand if it's rained in the last day or two. If not, however, you can use this website, wunderground.com/history, and put in the coordinates or the zip code of your sites to look up the historic rainfall over the past day or two. So that'll be your first step, to either think back over the past 2 days or look it up on this website to see if there's been any rain. Second, you'll label your bottles and fill your cooler with ice before you leave the house. I do recommend that you label your bottles first and let the ink dry for a minute or two before you put them in the ice, just in case, just so that you don't end up washing off those labels. And we have had issues with that in the past 2 sessions. So please make sure that you're labeling the sides of your bottle with the permanent marker that's provided and that you're giving it a moment to dry before you either get it in the water or put it in the cooler. All right, so once you've checked your rainfall and you've labeled your bottles and filled your cooler with ice, then you're, ready to head out into the field. So you're going to monitor all 2 or 3 sites that you've been assigned. And you're going to plan your route so that you get to each of those sites between 8 a.m. and 12 p.m. At each site, you will complete your visual assessment using the ArcGIS Survey 123, which is, again, that online form that I will show you in a little bit. You will, using the thermometer in your kit, measure the water temperature. And then you will collect 3 individual samples at each site. And there are 3 separate bottles, so a different type of bottle for each of these samples. So you'll use the sterile bottle to collect bacteria samples, the green capped bottle to collect a turbidity sample, and the clear capped bottle to collect a nutrient sample. Once you've visited all of your sites, you will transport water samples to the Watershed Center by 1 p.m. Once at the Watershed Center, you will complete a hard copy chain of custody sheet and record the temperature of your sterile bottle. And that's just so that we can make sure that that bottle has been kept on ice and the bacteria have not had a chance to grow during transport. And lastly, you will record your volunteer hours at thewatershed.org/hours. And this is mostly for our development team. It's helpful as a metric for donors to show how much community engagement we have. It also is helpful for you if you have any requirements to do community service. We will then are able to report on the amount of hours that you've donated. And so I'll go over that in specifics towards the end of the presentation. All right, so let's take a look at this online survey that I've mentioned a couple of times. In the kits that you've collected, there is a laminated half-page sheet that has a QR code on it. That's the same as the QR code on the screen right now. And there's also the link. So if you have your phone with you, scan that QR code and follow along. I'm going to be showing the web version, which is a little bit different. So it might be helpful for you to have it on your phone and go along with us so that you can see the differences. So I'm going to switch my presentation over to this form. Okay. All right, so Survey 123 is a product of ArcGIS, which is used for lots of mapping applications. And this is an online form which allows us to collect data in the field and then automatically transfer it to a map, which we can then use for data analysis. So the first part of this form is really basic. It's just like any other form. You're going to fill out your name, your email, the date that you're sampling, and the time at each site. And so you'll do one of these for each of your sites, right? So when you get to a site, you'll fill out your name, email, date, and time. Sorry. And then here you will write the monitoring ID. And so I have emailed you all of your site assignments already. And when I do that, it's a line of text that says, for example, Stony Brook, SBA, upstream of Titus Mill Road at, and then a set of coordinates. So what I'm looking for in this part of the form here, where it says monitoring site ID, I'm looking for that SBA code. So each site will have a 3 to 5 character alphanumeric code, and that's what we use across the watershed to identify our sampling locations. So please make sure that you're writing in that code and that you've got it written correctly from the email that I've sent you. That's how we'll know kind of which form goes with which site. The next question here is this your usual monitoring location? And so there are two options here. If you click yes, this is the usual location, that's all you have to do with this question and you can keep on going. But if you click no, show map to pinpoint your location, this map shows up. And so you'll do this at least on your first time out, but potentially if you have issues accessing the water from the same location week over week, you can select this more than once. So to use this map, you can use this little kind of homing beacon button on the side here. I believe on the phone, it's on the opposite side of the map to zoom into your location. And then you can and then you can zoom in even further to see where you're at and drop your point where you're closest to the water. So yeah, so please make sure that you're looking carefully. And when you do, if and when you do this, and like I said, you will do this on the first time you go out, make sure that you're zooming in as close as you can and dropping that point, trying to be as accurate as possible with where you're currently sampling that water. All right, so that's the mapping part. Next, we have weather conditions. You'll just mark down the current weather at your site. So sunny, partly cloudy, overcast, light rain, or heavy rain. I believe you can select more than one option should you need to. Last rainfall, this is where you will record the rainfall that you've either remembered or looked up on Weather Underground history. So within the past 24 hours, 24 to 48 hours, more than 2 days ago, or unknown if you weren't able to determine. All right. So that's kind of our like metadata. And now we're going into the actual visual assessment of the site where you're collecting your water. So first question here is water odor. There's a couple of options. The options are normal, anaerobic, sulfuric, sewage, petroleum, and other. Normal is kind of a green, streamy, maybe a little bit musty smell. Anaerobic is a little bit hard to describe, but if you smell it, you kind of know it. It's a it means there's no oxygen, so it's kind of a decaying, stale kind of a smell. Sulfuric will smell like kind of like rotten eggs. Sewage, I think uh, we all have experienced at some point or another what sewage might smell like. And petroleum is more of a chemical smell. And then if there's something else, you can always select other and type in your other smell here. And this question, again, can have multiple answers. Next up is watercolor. And this is a visual assessment of the entire water body, just looking at the surface. Does it look clear, green, blue-green, tea-stained, which is just like it sounds like. It kind of looks like tea, maybe a little bit brown. A little bit different from brown. This tends to be a little bit murkier and muddier. Sometimes the water may look yellow, and sometimes it may look gray or white. And we've seen this in places in particular that are downstream from like a quarry or something like that, where you're getting like stone dust, or potentially a construction site, something like that. So we have seen that occasionally. And then again, other is an option, and you can write in the colors that you're seeing. And you can select more than one if necessary. I will say that although this is meant to be kind of an overall view of the water from the surface, once you've collected your water samples and can look at the water a little bit more closely, you may want to adjust your selection based on what you're seeing in the bottle itself. Sometimes that color is a little bit easier to see once it's in a bottle and up close. All right, next is turbidity. And turbidity is the distance through the water column that you're able to see. So if you can see the stones on the bottom of the stream, that's pretty clear. Slightly turbid would mean that, you know, it's a little bit fuzzier. You can't quite see all the way through the water column, sorry. Turbid or muddy is kind of what it sounds like. It's muddy, like it's brown. You can't see through the water. There's a lot of stuff suspended in the water, and you're not able to see that through the water column. Milky kind of relates to that gray or white watercolor that we were talking about. Usually that means that there's a lot of stone, sediment, or something like that in the water. And then green pea soup could be an indicator of a harmful algal bloom, and I have some pictures of that in a minute. But that can also make it very hard to see through the water column. It just looks green and kind of soupy. And then again, other is an option here. For turbidity, there is only the option to select one, but with the other option, you can write in if there's something that doesn't fit under any of these categories. All right, next up is surface coating. And so this is, again, looking at the overall water body and looking at the surface to see if there's anything floating on the surface. So none is an option. Sometimes you'll see some foam, especially along the banks or in any kind of crevices of branches and twigs that have fallen into the water. Sometimes foam collects along those. Scum is, I think I have a picture of it in a moment, but scum is just a very thin surface coating. It's usually biological materials. Paint streaks, again, could be an indicator of a harmful algal bloom. These will look kind of green and they will have streaks to them. It'll look like someone just kind of dumped some green paint in the water and swirled it about. You might also have duckweed or other forms of vegetation that float along the surface, and that can be pretty common, especially in the summer when there's lots of sunlight and plants are growing. And then oil is an option. Oil can be introduced. It can be chemical oil, but it could also be naturally occurring. So you'll see that kind of iridescent rainbow sheen on the water. A good way to tell if it's natural is if you use a stick to swirl it around and it breaks into clumps, then tends to be a more natural oil. Whereas if you swirl it around with a stick and it clumps back together into one large clump, that tends to be more man-made. Of course, there's no option for indicating that here. You would just indicate that you're seeing an oil sheen. And then again, here you can choose more than one, and you have the option for other and to write in something else. All right, next up we have water movement. So you'll notice that each of these has a slash. So the word before the slash, oh gosh, what's the word I'm looking for? Would apply to a lake, whereas the word after the slash would apply to a stream. So the options are still or slow, ripples or moderate flow, waves or swift flow, and choppy or rapids. So again, if you're at a lake, you're going to look to the words on the left side of the slash. And if you're at a stream, you'll look to the words on the right side of the slash, just to give us an indication of how that water is moving. Aquatic vegetation, this includes macrophytes. So that is any kind of vegetation that has a leaf with roots in the water. It can also include duckweeds and other types of aquatic vegetation. You can select none visible, scarce, moderate amounts, or abundant. And so this is just taking into account all of the vegetation that you see across your site and characterizing kind of the amount of that. This next question here, the algal bloom presence, gets into our kind of watchdogging for harmful algal blooms that I mentioned earlier. So again, this is visually looking at the entire site and trying to assess how much algae there is. Your options are none visible, scarce, moderate amounts, and abundant. And I just want to show you here that if you click none visible, that's all you have to do for this question. But if you were to select scarce or either of these, and to change your selection, you just click the opposite option. And to remove your selection, you just click that, what is selected. So if you select anything other than none visible, you get this new question pops up. And this is to characterize the type of algae that you're seeing. So the options here are biofilm on rocks and on the bottom. Biofilm tends to be, in this area at least, it tends to be kind of brownish, and it makes the rocks look a little bit fuzzy. And this can be very slippery. So if you are going to a site that has a lot of biofilm, please be extra careful walking in and around the stream or lake. Okay, so biofilm is one of the options. Then there's filamentous and stringy algae, and I have a picture of that in a moment. But it's green and it looks stringy, and if you were to pick it up with a stick, you would see long strings of it. You can usually see it kind of flowing along with the water. If there's any movement in the water, you can usually see that filamentous algae kind of floating along with that movement. Then you might see floating globs, scums, and or pea soup. Again, I have some pictures to show you of that in a moment. And then suspected hab is an option here. And so again, with any of these questions that have a square here means that you can select more than one. So you might see biofilm, floating globs, and suspect a hab, and you can enter all three of those. That's fine. You also have the option to type in other and describe what you're seeing. Note here, if you suspect a hab, please report it to NJDEP right away. There is a link, once you've submitted this form, there is a link that will pop up for you to report your suspected hab. All right, so that's that. Next is water temperature. And I'm going to very quickly, let's see here, stop sharing so you can see me. See me?
Yes, we can see you.
Thank you. All right. So in your kit, you should have a thermometer. Your thermometer might be blue, like this one, and it might be purple, like this one. And so to collect the water temperature, what I would like for you to do is using the string that's attached to your thermometer, drop the thermometer down into the water column there's an end that has a bunch of holes in it. With the end with the holes down into the water, it's important that it's suspended in the water column, but not touching the bottom of the stream or lake. So you want to have this floating in the water for 1 to 2 minutes, just so that it can adjust to the temperature there. And then you're going to pull it out of the water and holding it by the top. You'll read the number on the thermometer itself and read just as to the closest line as possible. You don't need to kind of make up numbers in between the lines, just at the closest line that it's at. I do want to just mention that when you're reading, do not hold the end of the thermometer here. This will start to change that temperature to warm it up with the heat of your hand. So when you're reading, please make sure that you're holding it from the top and just reading the number here without touching the bottom. All right, so that's the thermometer. Go back to this form. So once you've read your temperature, you'll type in your water temperature here. And then this is just to let us know whether or not to expect water samples. So if you went to your site and collected water samples, you'll select yes. Please do try to collect your water samples. We're not able to do the lab analysis and collect the data that we need without the water samples. That being said, if you feel unsafe and if there's a really raging algal bloom going on and you don't feel safe collecting that water, you can click no and just submit the visual assessment. That's fine. Additional comments, this is just what you see going on. Is there construction going on? Is it or do you see a lot of geese around? Anything that you feel that might be noteworthy, you can add into these comments here. And then finally, we have 3 picture options. So the first is an image of the overall water body, just because we're not going out to these sites. So it's helpful for us to see kind of what you're seeing when you approach your site. So when you're doing this on your phone, you can either take the picture ahead of time and upload it once you get to this part in the form, or I believe you have the option to take the picture directly from the form. If you have bad reception at your site, which a couple of the sites can be a little bit spotty, I would recommend taking the picture ahead of time and then waiting until you get back to your car or back to a place where there is sufficient connection for you to upload the photos and submit your form. The second photo here is a close-up of the algal bloom, and this option will only pop up if you don't have none visible selected in this algal bloom presence. So you see here I've selected none visible, and now that second photo option is gone. If you select scarce, moderate, or abundant, either of those 3, this close-up of algal bloom option pops up and will ask for a picture here. And then the last is just an additional view and is an optional photo if you would like. Once you've filled all that out, you'll click submit. Let's see if I do that now. It won't let me. Anyways, once you click submit, it'll pop up with a screen that says, thank you for submitting your data. If you have suspected an algal HAB, please click this link to report it to DEP, and you'll report that right away to DEP. Alrighty. Oops. Let's switch back to my presentation just a moment. Sorry, lost my place. Okay, so any questions on the Survey 123 form before we move on?
Yeah, I just have a question about one of the initial questions on the form.
Sure.
So I just wanted to confirm about what you said about how on the first day you fill out that you're at a new site and the
Yeah,
pinpoint location of it. So you do that for all 3 locations on the first day,
correct.
and then in the following weeks, you just click yes and then submit time, and that's it.
Right, and the rest of the form. But yeah, for the rest of the times, unless for some reason you're not able to access the water at the exact same location, um,
Mm-hmm.
if that is the case, then you would click no and redo the point. But if you're going to the same spot for the next 5 weeks, just submit the map point once on the first week, and then you can select, yes, this is the usual location going forward.
Okay. So it wouldn't
And that's
matter like in what sequence you would go to each location each week. It can be like different locations, different times each week.
absolutely, yeah, that's fine.
Oh,
Yeah,
okay,
as
thank
long
you.
as all 2 or 3 of them are within that window, yeah, that's fine.
Okay.
Okay. And the map thing is just so that we can confirm that you're going to like the right location, or if we need to switch up the access to that site for some reason, we have the new coordinates so we can update our information about where to access the water in that site. Okay, so let's talk about harmful algal blooms, unless there are any other questions about the form. No? Okay. Harmful algal blooms. I'm sure you've heard about them this summer because we kind of had a happy summer. Harmful algal blooms is a bit of a misnomer, actually, because it is caused by a bacteria, not an algae. So cyanobacteria are an essential photosynthesizing bacteria that are ubiquitous, which means that they are all over the place in pretty much any type of aquatic or even marine environment. There are these photosynthetic bacteria. They're not necessarily bad on their own, but when there is too much cyanobacteria, that's when we can start having trouble. So HABs, or harmful algal blooms, are cyanobacteria blooms that produce toxins. So not all cyanobacteria will bloom like this, and not all blooms will produce toxins. It's a little bit niggly, but yeah. So some, but not all cyanobacteria produce toxins, and those toxins include cylindrospermopsin, anatoxin A, and microcystin. Toxins can be produced, these toxins are produced when there's a large amount of these cells in the place, the concentration of the toxins can increase. And then if they come into contact with humans or animals, they can have a bunch of different responses depending on that concentration. Responses can include something as simple as skin irritation. If it happens to be ingested, it can include digestive upset and flu-like symptoms. And it even at very high concentrations, it can even lead to organ failure and even death. Um, this is super unlikely, but we have seen it occur, especially in pets. Sometimes, someone will have their dog playing at the edge of a stream or a lake, and the dog will drink the water. And if there's enough toxins in there, that can cause some issues. So that's one of the main reasons that we are doing this monitoring, is to keep an eye out for the occurrence of these harmful algal blooms and to notify DEP as soon as something seems like it might be heading that way, so that we can prevent any, especially deaths, but of course, any illnesses due to these toxins. So how do you even make a HAB? Because these bacteria are photosynthetic, they require nutrients and they require sunlight. And you put that together in a slow moving body of water, such as a lake or a stream with low flow. And that's when you get a HAB. And so the nutrients are introduced through runoff. So if there is a large, when it rains, the water will pull any nutrients on the land into the water. So this can include nutrients from fertilizers, it can include things from streets, so oil from cars and things like that, but it can also include fecal matter from waste and nitrates from fecal matter and stuff like that. And so that will increase the ability of that bacteria to reproduce and to photosynthesize, and then we get these blooms that we're talking about. So the best way to prevent a HAB is to reduce stormwater runoff and to allow that water to infiltrate through the ground where a lot of these nutrients get filtered and don't end up in the lakes and streams that are then sitting there and allowing that bacteria to eat it up and reproduce. So let's take a look at some pictures of what harmful algal blooms might look like so that you're aware. This first picture here, and all of these pictures are from the NJDEP website. You can look at their HabPics website. The links are here. So this is what we would describe as a pea soup looking site. You can see the water is very, very green. It doesn't look like it's sitting on top of the water. It looks like the water itself is green. So that's what we would call kind of a pea soup looking site. Then we have what we would call the paint streaks. So this does look a little bit like it's on the surface in this case, but you can see the streakiness of it. You can see the movement of the water through those streaks. So this is what we would call kind of a paint streak looking site and can be an indicator of a harmful algal bloom. These first two, the pea soup and these green paint streaks, are pretty definite indicators of a harmful algal bloom. Whereas this next picture here, this foam, it is green. The water underneath it looks pretty green, but is less. So the first two, the pea soup and the paint streaks, are like almost 90% sure that that's a harmful algal bloom. This one, I would say, is maybe a 60% could be a harmful algal bloom. So this is the foam where you're seeing, you can see it kind of clumps up against the twigs or against the bank of the stream or the lake. And this foam is very green. This is another possible harmful algal bloom. Again, not super definite, but you can kind of see these green dots in the water. That's a potential harmful algal bloom. And we do have these occurring. So 2 years ago, we had a harmful algal bloom happen in the Millstone River. So as I was saying before, harmful algal blooms are more likely to occur in places where there's not a lot of flow, where the water is still, where the nutrients have a chance to gather, and the water has a chance to heat up from the sunlight and produce that environment for growing cyanobacteria blooms. So for this to occur on the Millstone River is pretty unique. That's a large kind of flowing body of water. But you can see here the difference between the harmful algal bloom happening in the Millstone and the Beeden Brook on the right, where the water is clear. There's no harmful algal bloom happening here. So this is just a good picture to show the distinction between the two. You can really see that green water there. And this is another picture of that same harmful algal bloom in the Raritan in 2022. This is the confluence of the Raritan River. And again, you can see that water is really green, whereas the Raritan is not that green. So some ideas of what to look for when you're trying to suss out if this is a harmful algal bloom. Here's a new picture. This is what we call duckweed. It's an actual macrophyte. It looks like it's on the surface. You might kind of think of this as paint streaks, but if you were to look closely at this, you would actually see little tiny leaves with stems into the water column. And so this is a type of aquatic vegetation called duckweed and is not a harmful algal bloom. So I'd like to make this distinction because we do get occasional false reporting where people see a lake covered in duckweed and think that it's a HAB, and really the water underneath is fine and it's just that duckweed on top. And then this is another example of what could constitute kind of a false reporting of a harmful algal bloom. This is a picture of what we call filamentous or stringy algae. I realize this isn't the highest definition photo, but you can see the streaks of algae in there. If you were to pick this up with a stick, you would you would see the strings of the algae coming out. You can usually see the algae flowing with the water, and the water around it is clear. You can still see through to the bottom of this water despite there being tons of algae here. So this is a true algae and is not a harmful algal bloom. So here's a quick test for you. Which of these would be a suspected HAB? And you don't have to say it out loud, but just think in your head. If you're looking at these two samples, which one would you think is closer to being a HAB? And if you're thinking that the one on the left is more likely a HAB, you would be correct. The one on the right is a sample that has lots of duckweed in it. And you can see those stems. You can see the little leaves at the bottom of that bottle, and then you can see the stems hanging down from the water. That's just an aquatic vegetation called duckweed and is not a harmful algal bloom. And as an added note, samples like these will wreak havoc on our analytical equipment. So we do ask that you try as hard as possible to not get vegetation and debris into your water samples. The analytical equipment that we use has very small tubes and syringes and things like that. And so any kind of debris in the water can clog those up and cause a real issue for us. So when you're collecting your water samples, please be as careful as possible to only get water and not get a bunch of extra dirt and vegetation. So then I wanted to show you this sample as well. If you look closely, you can see little green dots, and those are clumps of those cyanobacteria that are starting to clump together. And that's if you pull up a water sample and you happen to see these green clumps in there, that's a pretty good indicator of a harmful algal bloom as well. So some of these questions on the visual form might be easier to answer once you've collected your water samples and can you look at the water a little bit closer. All right, so if you do see something like that, we'll ask that you please report that directly to NJDEP. So again, once you've submitted your Survey 123 form, there will be a link to report to DEP should you need to. Once a harmful algal bloom has been, a potential harmful algal bloom has been flagged, the watershed staff will go and do a phycocyanin measurement. Phycocyanin is a pigment found in chlorophyll or in photosynthesizing organisms. And it helps us to correlate with cyanobacteria cell counts. So that's what we would do at the watershed, is take a phycocyanin reading and determine if there might be a high enough cell count to warrant further investigation. And then we would also report it to DEP, where they would do a more in-depth analysis to take samples to actually count cyanobacterial cells, and then also to quantify the toxin levels to determine just how bad of an algal bloom is occurring. So I do want to point out that that is not analysis that we're able to do at the Watershed Center. We're only able to flag that there could be a HAB happening, and then DEP will do the more in-depth analysis. They will also report their findings on this nj.gov/DEP/HAB website. And I have a screenshot of their dashboard here. So these are the sites. This is an older screenshot from June, but these are the sites that they visited and the results of their analysis on those water samples that they collected. So the blue ones are watches. There's not a full-blown bloom happening, but it could happen. Like, conditions are right, it's getting there. Advisory is kind of the next step up, warning, and then danger. So if you go to a site and you see any of these signs, these are from DEP. They have been out there to test the water already, and they can let you know what level of danger kind of you're in. So you can use the link here to view their dashboard online and check your sites before you go if you'd like to. And if you report a site, it should show up here within a week or so. All right. Any questions about harmful algal blooms before we move into collecting water samples? All right, we'll move in. So to collect your water samples, you're going to need your sampling kit. And I sent it out in emails, but hopefully you've already gotten your kits. They are available at the Watershed Center. You go to the side entrance of the watershed, and there's a cabinet outside, so you can grab your kits. It's outside of the building so that you can go at any time, whether the building is open or not. But for your information, the building is open from 9 to 5, Monday through Friday, and 10 to 4, Saturday and Sunday, except for like holidays and occasional private events. But regardless of the hours of operation, the bacterial cabinet is at the side entrance and available to you at any time. Inside that cabinet, you will find a bunch of these gray kits. Inside the kits, you should have maybe not this many, but some of these sterile bottles, a bag of gloves, a thermometer, a Sharpie, the laminated quick guide with a QR code for the form, and these additional sample bottles. So you'll have the green capped bottles that we use for turbidity analysis, and the clear capped bottles that we use for nutrient analysis. And so on these bottles, before you leave for your site, you're going to label with your name, the site code. So again, that's that 3 to 5 digit alphanumeric code. So your name, the site code, and the date of sample. And I ask that you please write that with the permanent marker that is provided on the side of the bottle, not the top of the bottle. And I say that because we reuse these bottles week over week. And so we use alcohol to remove the Sharpie, and it's much easier to do that on the side of the bottles. For some reason, the plastic that the bottles are made of, it's easier to remove those labels than it is from the plastic that is used for the caps of the bottle. So please make sure that you're writing those 3 things, your site code, your name, and the date of sampling on the side of your bottle. All right, now we'll go into the one-handed aseptic bacterial sampling technique. And so this is particularly important for your sterile samples. Again, so that we're testing the bacteria that's actually in the water and not the bacteria that is on your hands or in your car or in the cooler, etc. So again, first you'll label your bottle with your name, the site code, and the date. You're going to use one of the gloves provided and put it on the hand that will be removing the bottle cap. You'll carefully uncap the sample bottle and hold the cap in your gloved hand, being extra careful not to touch the inside of the cap or the inside of the bottle. You'll submerge the bottle about 6 inches below the surface of the water, again, avoiding any vegetation or algae, and fill above the 100 milliliter mark. But you do want to leave a little bit of space in the bottle so that we are able to shake it up and mix it up. So above that 100 milliliter mark, but not all the way up. Generally, if you're able to fill the bottle up to the shoulders of the bottle, that should be enough. Then using your gloved hand, you'll recap the bottle and immediately put that sample in the cooler surrounded by ice. And again, here, it's important that it is loose ice and not just an ice pack. We want to make sure that that sterile bottle of bacteria is completely surrounded in the ice and able to be cooled uniformly so that the bacteria does not have a chance to continue to grow in the bottle between the time that it is sampled or picked from the site and the time that we are able to analyze it in the lab. So I'm just going to stop sharing my screen and do a little demo here. Okay. So you'll have your sterile bottle, and these are the clear bottles with the white cap. They're the largest of the 3 bottles that you have. You're going to take your glove And you only need to glove one hand unless you're worried about a potential harmful algal bloom. You only need to use one glove. You will use the gloved hand to remove the cap of the bottle. And then you'll hold this in your gloved hand, being very careful not to touch the inside of the cap or the inside of this bottle. So you'll hold this in your gloved hand, and then you'll submerge your bottle. If this is the surface of the water, you want to go 6 inches below the water to collect your sample, right? Surface water could be a little bit different. There can be a lot of debris and stuff. So we want to make sure that we're not sampling just the surface water. We want to get at least 6 inches below the surface. Scoop up that water. If you look closely at your bottle, and hopefully you've got your kits with you so you can look at one closely, you'll see there's a line right about here that marks the 100 milliliter line. So if you fill your bottle up to the shoulders of the bottle, which is this, this step right here, that should be enough water for us to do our analysis. For these samples, we need exactly 100 milliliters. So if your sample is way below this line, we're just not able to do the analysis and get the E. coli data that we need. So please make sure that you're filling up to the above the 100 milliliter line. Okay, and then once you're done filling your sample, you'll close the bottle with the gloved cap and put this right into your cooler filled with ice. Okay. When you're collecting your samples, and if there's a lot of debris on the surface of the water, you might want to use your hand to kind of move some of that debris out of the way before you dunk your water in so that you're not collecting a bunch of debris as well. All right, that's the one-handed aseptic sampling technique. Then put my presentation back up. So the only time you absolutely need to use a glove is when you're collecting this bacteria sample. Again, you'll label the sample first, put the glove on the hand that will remove the cap, uncap without touching the inside of the cap or the inside of the bottle, submerge the bottle about 6 inches, avoiding any debris, fill above the 100 milliliter line, but leaving a little bit of airspace, recap the bottle with your gloved hand and put the sample bottle in the cooler surrounded by ice. And then you can proceed to your other sample collection. It's much the same, but you don't need to worry about gloves in this case. You'll label your name, the bottles with your name, site code, and the date. In this case, because these are not sterile bottles, we want to rinse them 3 times with the water at the lake or the stream. So that just means you'll dunk the cap and the bottle 3 times in the water, dunk it in, pour it out, dunk it in, pour it out, dunk it in, pour it out 3 times before you collect your water sample. So it's important to do that with the bottle and the cap for each of those. Submerge the bottle about 6 inches, again, below the surface water, avoiding any vegetation or algae or any other kind of debris that might be in there. And then you're going to fill more than halfway, as full as you can, leaving just a tiny bit of airspace so that we can shake them up and mix. Recap the bottles and then immediately put them into the sample cooler, into your little cooler bag surrounded by ice. Again, with loose ice, not just an ice pack, we want to keep that temperature stable through all around so that the parameters that we're measuring are not changing during transport. So at each site, you're going to fill one of the large green capped tubes and one of the smaller clear capped tubes. So at each site, you're going to have 3 separate samples. To deliver the samples, you're going to go to the side entrance of the watershed center. You will make sure, please do make sure that your sample bottles are all closed tightly. And I will here make a note that the green capped bottles, there are two different kinds and the caps are not interchangeable. So just please make sure that your cap actually does fit on there and is nice and secure so that we're not spilling water. And we've had a couple of times where that's happened and then we don't have enough water to like do the analysis that we need to do. So please make sure that your samples are labeled and closed tightly. Then you'll fill out the chain of custody data sheet. There is a binder inside the cabinet with a chain of custody sheet, and you'll fill out the site code, the number of bottles that you collected at each site, the date, I believe, time that you're delivering, and your name. Inside the cabinet, there is also a laser thermometer, and so you will use that to take the temperature of your sterile bacteria sample bottle. And so there's also a cloth in there that you can wipe off the side of the bottle and use the laser thermometer to take that temperature and then write that on that chain of custody data sheet. Then we have a large green cooler that will sit on the floor across from that cabinet where you picked up your supplies. And so you will settle your samples into the ice in that cooler. And then you'll take your own cooler back with you. So you can dump the ice out into the grass right there and take the cooler back with you to use the following week. And then you'll also pick up however many sample bottles you need for the following week. So if you only have 2 sites, then you need 2 sterile bottles, 2 green capped bottles, and 2 clear capped bottles, for example. We'll also have additional gloves in the cabinet if you need them. I would ask, though, that you use the gloves sparingly. We try not to produce garbage unless we need to. So unless you do need at least 1 glove to do your sterile sample each week, but hopefully that is all you need unless you have a super heavy site. Okay, so this is what that bacteria data sheet, that chain of custody sheet looks like. And so what you will do is you'll write your site ID, again, using that 3 to 5 digit alphanumeric code. You'll write down the number of bottles that you collected at that site, which at each site should be 3 samples. The temperature of your sterile bottle. Again, this is to make sure that so that we know that it has been kept on ice and the bacteria have not had a chance to grow during transport. The time of delivery, that's so that we know which samples need to be analyzed first. And then sign off that, yes, I have collected these and I am delivering those samples. And so you'll fill out 1 line for each of the sites that you visit. So if you've only visited 2 sites, you'll fill out 2 lines. And if you've visited 3, you'll fill out 3 lines. Inside the cabinet, you'll see these bins with additional bottles, gloves, and then this is the thermometer. You'll just point it at your bottle, pull the trigger, and then let go, and it should hold the temperature and long enough for you to write it down on the data sheet. These are just like the thermometers that everyone used during COVID, so hopefully it's pretty straightforward. All right, so what happens once you've delivered your samples? We bring them into the lab. We bring them into the lab and we add what's called Call Alert. It's a reagent that kind of feeds the bacteria in the sample and allows it to grow at a known rate. We incubate them in these trays overnight. These trays allow us to subsample the 100 milliliters, incubate them overnight, and then we will take a look at them after they've incubated. If the water in these cells turn yellow, that's an indicator of total fecal coliform. So that's an indicator of bacteria that is present due to fecal matter. And then we'll take a look at it under a UV light. And if it's yellow and fluorescing like these are, that is an indicator of E. coli. So we will count up the number of cells that have changed colors. we will count up the number of cells that have fluoresced, and then we can determine exactly how much bacteria or E. coli is present in that water sample. And we compare that number with the New Jersey water quality, surface water quality standards. So these are numbers that are provided by DEP. The standard for a single sample is 235 colonies per 100 milliliters of water. And the reason that we do these 5-week sessions is because the standard is actually based on a geometric mean of 5 samples taken within 30 days. So that's 126 colonies per 100 milliliters of water. So we take the numbers that we've counted of the cells, and we convert that to these number of colonies, and we compare them to these standards and decide whether or not the site has passed or failed. And this little diagram on the right is just showing that E. coli is a type of fecal coliform bacteria, which is a type of total coliform bacteria. So we're really focusing in on just this one type of bacteria that we know causes distress in humans and animals. And so once we've done that and we've compared the data against our surface water quality standards, we then publish it to our StreamWatch dashboard, which is available on our website. If you go follow this link, thewatershed.org/bacterial action team, and scroll down to the bottom of the page, you'll see this dashboard. And this is actually updated weekly with the data that you collected. So you can go after this Sunday. Usually we have the data up by Tuesday or Wednesday. So on Tuesday or Wednesday, you can go, you can find your site, and you can see what each of the parameters that we measure has done over certainly at least on Sunday when you measured, but if there's been a volunteer measuring your site or monitoring your site for previous weeks, you can see how it's gone over those previous weeks. So that's what we do for the bacterial action team, and then that is added to the data collected from the chemical action team and the biological action team, and we produce our clean water report cards once a year. So the clean water report card, the 2023 clean water report card was released in, I think it was June. And so that takes the data that's collected by all 3 teams over the last 5 years, combines them all, compares it to the water quality standards, and then we give a rating of excellent, good, fair, or poor for each of the parameters for each site. And then we also combine parameters to give an analysis for each subwatershed and municipality. So if you scan this QR code on this slide, you can visit our story map that has all of the data that we've collected over the past 5 years, kind of boiled down into this report card that's hopefully understandable. So that's where your data ends up. We do also report our data to DEP and the EPA so that they can use this data for regulatory purposes. All right, so to sum up, I just want to review your weekly routine. First thing you're going to do is either remember back or check on weatherunderground.com/history the local rainfalls for the past 24 and 48 hours. You'll label your bottles with, again, your name, the site code, not the name of the water body, but the site code, and the date of the sampling, and fill your cooler with ice before you leave the house. Then you're going to visit all of the monitoring sites on your itinerary and plan to do so before 12 p.m. At each site, you'll complete the visual assessment using Survey 123, that online form that we went over. You'll measure the water temperature using your provided thermometer, and then you'll collect 3 samples at each site: a bacteria sample, a turbidity sample with the green cap, and a nutrient sample with the clear cap. You'll then transport your water samples to the watershed center by 1 p.m. And I will just add an asterisk here that if you happen to be running late, which please try not to, but if you happen to be running late, we will be in the lab at the watershed center conducting these analyses at 1 p.m. So if you happen to come late and the green cooler is no longer by the side entrance, you can bring your samples directly into the lab so that we can get them received and start analyzing them right away. So you'll transport your water samples to the center. You'll complete the hard copy chain of custody sheet and record the bottle temperature of your sterile bottle. And then when you get back home, you'll record your volunteer hours at thewatershed.org/hours. So this is just a reminder to log your hours. If you have not done so, please make sure that you contact Eve and complete your volunteer forms. And then use a simple online system to record the hours. That's the link again, thewatershed.org/hours. 2 things to be aware of when you go to complete your hours online. Please be consistent with how you record your name. So if you're Elizabeth, but you're sometimes Beth, just kind of pick one and stick with it so that we know who you are, and we can make sure that those hours get added to the right person. And then all of the time that you spend working on this and on StreamWatch can be included in your hours. So that includes the time that it took for you to go pick up your sampling kit, if you've already done so, the time that we've spent this morning going over this presentation, driving to and from your sites and to the Watershed Center. Any and all time that you spend on behalf of StreamWatch and the Watershed Institute can be included in these hours. And I think that's all I have for you. So I'm going to stop sharing and see